want to welcome everybody to the first of a series of seminars that we're going to be running that are really dedicated to discussing life sciences and how our life science customers can leverage Google Cloud. So we'll just do a round of introductions. So my name is Vincent Beltrani. I am a computational chemist, and I'm also a customer engineer here at Google Cloud. I work out of the New York City office, and I am on the healthcare and life sciences team. In particular, my primary focus is on life sciences and things like scientific computing, including HPC and GPU computing, AI, ML, and big data and its use cases really across the sciences in general. So it's really great to see you all, and I'll pass it off to Tim for a quick introduction. You're on mute, Tim.
Sorry, there you go. Thanks very much, Vincent. Thanks very much. Yes, great to be here today. My name is Tim Elwell, and I lead the advisory services group for Quantiphi. Quantiphi is a AI ML first digital engineering company. We're partnering with Google. I've been in the industry for about 35 years or so, and am really focused on being able to support from a consulting perspective what clients' needs and trying to translate those needs into a requirements document and to be able to create opportunities for success. I'm going to pass it over to Vishal.
Sure. I'm Vishal Pagiripalli. I'm a machine learning solution architect. I work within the healthcare and life sciences business unit in Quantiphi. I also lead a research team within the business unit, and our primary focus is building capability within a lot of our strategic areas, which are like radiology and pathology and a lot of life sciences work which we are doing right now. Yeah, happy to be on this call.
Hi everyone, my name is Sofia Mosho. I am a PhD on computational physics, and I'm also a senior machine learning engineer with Quantiphi. What I work on is custom machine learning workloads for applied research purposes, and I'm helping clients in healthcare and life sciences put together and orchestrate their machine learning operations workloads. With that, I will pass it on to Vincent to kick off the main part of the session.
Great. So I want to thank you all for coming to the first of a series of our seminars that are all focused on life sciences. As a customer engineer and as a computational chemist, I've had the opportunity to work with many of our life science customers, and they're doing amazing work. The goal of these sessions is to demonstrate how you can take all of that great scientific work that you're doing today and combine it with the power and the scalability of Google Cloud. I'm going to spend just the next 10 minutes talking about some of the science and technology that we'll discuss, and then I'm going to pass it off to Tim, Vishal, and Sophia to do a deep dive. So hit identification is a critical stage of the drug discovery process, and often that's facilitated by enumerating large virtual libraries to be used downstream in things like virtual screening. The size, complexity, and the diversity of your virtual libraries plays a key role in increasing the chances of finding a potential drug candidate. Constructing virtual libraries can be done in a variety of different ways, using reaction schemas, scaffolds and templates, graphs. There's all sorts of different methods for generating these libraries. You normally also include filters or requirements to ensure that those compounds have some novelty and that they're synthetically feasible. The one thing that all of these techniques have in common is that they are computationally intensive. So many of our customers have used their specific subject matter expertise to tailor their own pipelines, but they're running them on their local machines where they're limited by the capabilities and the availability of their own hardware. And since the size of drug space is huge, something like 10 to the 60th power, scientists need to unlock their ability to do these types of computations and these types of calculations in a scalable and an elastic way. So instead of running pipelines on local hardware, researchers can take advantage of the scalability of the cloud. You can also take advantage of managed services, which are services that manage resources and the infrastructure for you. So that means instead of worrying about virtual machines, you can actually worry just about the science. And instead of having to worry about setting up storage or standing up your own database, you can instead use our cloud storage service and BigQuery, which is a managed data warehouse. Cloud Data Flow is another managed service. It's a managed data processing engine. You can work with large numbers of multiple data sets in batch and even in streaming mode. You're only responsible for the functional parts of your code, and then you hand that code off to Dataflow, and Dataflow is responsible for determining where to run the code, how to run the code, and importantly, how to scale it optimally across multiple workers for you. So we've used this type of architecture to calculate molecular fingerprints across 1 billion molecules in a matter of hours. Later, Sophia is going to discuss an architecture for using Cloud Data Flow to create those large virtual libraries. Another focus of the drug discovery process includes leveraging all of the information that's already been published by other scientists and researchers across the world. Of course, staying on top of the literature is basically impossible. Since the first published paper in 1665, in the Philosophical Transactions of the Royal Society, there have been over 50 million peer-reviewed papers published. The amount of data out there is enormous, it's growing, and that's just publications. On the next slide, you'll see that in reality, there are many, many other sources of chemically and biologically relevant information scattered everywhere. For patents, clinical trials, conference papers. Even the archive now, as of 2022, has over 2 million articles. And you likely have a ton of your own data that you've collected over time in the form of IND applications, laboratory notebooks, and internal documentation. So if you wanted to build a machine learning system to extract information from all of that scientific literature, a key ingredient is the ability to parse those documents and recognize and identify compounds and reactions that are mentioned throughout scientific texts. Quantiphi has used Google Cloud technologies to build out a state-of-the-art system to do just that. And Sophia is going to walk us through that architecture in just a little bit. We're also going to talk about the applications of AI and machine learning to the challenging problem of developing vaccines, therapeutics, and diagnostic tests for SARS-CoV-2. So SARS-CoV-2, as you know, is an RNA virus with a high mutation rate. Mutations in the virus change the profile. So for example, making it more contagious. This is an image from a paper published in Nature that analyzed different mutations. You can see the red section here is the amino acid sequence for the spike protein. So as you can see, there have been numerous variants, numerous mutations across variants. And this is really just a sample from 2021. So the real question is, if you were designing therapeutics, or if you were building diagnostic tests to identify COVID-19, how would you try to explore their efficacy against potential future mutant variants? So by now, you've probably heard of AlphaFold, a deep learning-based protein structure prediction tool developed by Google's DeepMind. Today, Vishal is going to talk about using AlphaFold to predict the structure of the mutated SARS-CoV-2 protein and what that kind of architecture looks like on Google Cloud. I'm not going to get too much into the details of AlphaFold itself, except to highlight that the system was trained across the entire protein data bank and many, many more data sources. Training a model of this size with this degree of complexity on traditional hardware would take months. But AlphaFold system took just over a week to train because DeepMind leveraged cloud TPUs, or cloud tensor processing units, which are purposely built hardware on Google Cloud for machine learning, training, and inference. One TPU can provide up to 420 teraflops of machine learning processing power. And when they're connected into a mesh or a networked architecture, a TPU pod can exceed 100 petaflops of machine learning power. Those TPUs and those TPU pods are available for you to use today. It's pretty simple. You can either spin up one of our deep learning virtual machines, or probably even easier, play around with TPUs in one of our managed notebooks, or even in Colab. So most of the time when we talk about machine learning, we focus on the model and the code. But what I've highlighted here is the real perspective from a Google paper published in 2015 called A Hidden Technical Debt in Machine Learning. I've copied it directly in order to be able to read the caption. So figure 1, only a small fraction of real-world ML systems is composed of ML code, as shown by the small yellow box in the middle. The required surrounding infrastructure is vast and complex. So that's where Vertex AI, our end-to-end machine learning framework, comes into play. It manages all of that surrounding infrastructure and all of that complexity for you, along with also managing and supporting your machine learning code. So in the next slide, you'll see that Vertex AI provides all the tools for every step of the machine learning workflow across different model types and for different levels of machine learning expertise. And that's really, really important, because if you really want to accelerate AI innovation, you need a platform that can both help build expertise in novice users and simultaneously also provide a seamless and a flexible environment for your machine learning experts. In your Vertex AI pipelines, the first thing you do is ingest data, validate it, and then prepare it with some types of transformation. Then you create and train your model, evaluate that model for efficiency, and deploy it for making predictions. With Vertex AI, all of that is done in one central place. For model training, you have options like AutoML and custom training. With AutoML, you don't write any code. Vertex AI takes care of identifying the best model for the task based on the data and the data type, images, tabular data, even video. For other use cases where you want more control, you can use custom models. That's great because you can use code that you've already written or code you want to write yourself using frameworks like PyTorch and TensorFlow. And once those models are deployed, Vertex AI manages the prediction endpoint for you and scales it on demand as part of the managed service. There are also tools to monitor your model over time to help determine when it needs to be retrained. There are also a few other features of Vertex AI that I'll just mention quickly next. So like I mentioned before, you can use your favorite machine learning frameworks, be it TensorFlow, PyTorch, scikit-learn, and so on. Every aspect of Vertex AI is pre-configured to work with accelerators like GPUs and TPUs, and there are built-in visualization tools like TensorBoard. There's also a metadata management system that catalogs every artifact in your machine learning experiments, so they're all audited, and you can go back and repeat an experiment at any time. I think my personal favorite feature of Vertex AI are our managed notebooks, because as a scientist, I find them to be so useful. With the click of one button, I could spin up a Jupyter notebook, have Git or cloud repository integration in order to version my code. I can connect directly to the entire cloud ecosystem. So for example, my common working pattern is spinning up a Jupyter notebook, pulling in a library like RDKit, and then getting to work with data that lives either in BigQuery or it lives in cloud storage. It's really an easy way to get to work and start experimenting. So with that overview of some of the science and some of the technology that we're going to talk about today, I'm going to pass it off to Tim. Thanks.
Hey, thanks very much, Vincent. So as one of the consultants in the room, I'd like to frame the issue with a problem statement that needs to be addressed. So firstly, life sciences and more specifically big pharma manufacturers face a number of intractable problems that are present in the drug discovery process. And as a business person, our goal is to create products and solutions that are better, faster, and cheaper. However, as a scientist and as a fellow researcher, we can't continue to do things the same way and expect different or improved outcomes. So over the next couple of decades, or certainly over the last several decades, the cost of drug production has almost tripled. I remember when I was first researching this space and identified that the cost of a drug to market was about $500 million. And I thought that that was outstanding and impressive by itself, but now we've almost tripled that. The direct cost associated with bringing a drug to market is around $1.4 billion. And then when you take into consideration that it takes on average about 12 years to be able to bring that drug to market, the lost opportunity, the time value of money, lost opportunity cost is about another $1.2 billion. So generally, we're talking about $2.5 to $2.6 billion associated with the high development cost by itself. And then when you throw on top of that another $300 million to $500 million associated with things like, studies that require new indications or new formulations, or even when you're taking a look at dose strengths and regimens, or even being able to comply with regulatory agencies like the FDA to be able to ensure that patients are safe, all those types of components continue to further drive up that cost almost to $3 billion. So cost aside is obviously an issue, but there's other issues associated with being able to bring that drug to market that are problematic. For instance, low clinical trial pass rates are indicative in this particular space. There's been a number of studies that have looked at anywhere from less than 12% to 14%. Of the drugs are successfully able to pass clinical trial requirements. And then when I take a look at, for instance, finding and retaining candidates, for a particular study, a staggering 86% of all clinical trials fail to recruit the sufficient number of patients that are required for that particular study. Overall leads to lower or slower research processes, as well as the ability to bring patients life-saving medications, drugs. 80% of the drug clinical trials don't meet the enrollment timelines. 1/3 of phase 3 clinical trial are terminated due to enrollment difficulties. Lastly, about 7 out of 8 compounds that enter into the clinical trial process will fail. All of those things being said, obviously impacts the overall drug cost, but there has to be a better way. So the next two speakers are going to be speaking about how AI and ML are perhaps part of that answer. And with that, I'd like to introduce you to Sophia, who's going to discuss how ML will be able to improve the molecular translation component of that process. So Sophia?
Thank you, Tim. So the first use case we will talk about is molecular translation. For that, we have a molecular input image, like you can see in the left-hand side, that we want to translate into its underlying chemical structure. This can help us to interpret all chemical images and convert them back to their underlying chemical structure, that they might lie within all these volumes upon volumes of papers that Vincent told us at the beginning of the session, that there are plenty out there. When this process is automated and applied to a large number of scientific papers, it can help us make collective chemical research much more accessible, as we won't have to go physically through every single one of all those papers. And also it can help us identify novel trends by mining large data sets. But maybe more importantly, it can help us speed up the R&D efforts by identifying and excluding previously published molecules from downstream drug discovery cycles. Next, we will discuss the solution we put together for this. And we put together a flexible modular solution. If you exclude the pre-processing and post-processing steps that are part of pretty much any workflow and pipeline, our solution consists of 2 machine learning models, 2 deep learning models that we orchestrated to work together. The first one is that of an encoder, and the second one is a decoder with an attention mechanism. Next, we will take a closer look at the encoder parts. We played around and we implemented multiple different types of encoders for this particular workload. And what an encoder does is that it gets an initial input image and it passes it through multiple convolutional layers until at the end we get a lower dimensionality embedding of the initial image, original image. For this particular case, we put together implementations for EfficientNet B and EfficientNet V2 with different sizes of models. But what I would really like to emphasize here is that we put together a custom implementation of a vision transformer as the encoder part of the workflow. And this, if you're not familiar, it is a very much state-of-the-art computer vision algorithm. Next, we will take a closer look at the decoder. So the decoder now will take as input the image embedding that the encoder computed for us, together with the tokenized version or a broken down version of our target labeled output text into images or particular chemical structures. This is a supervised machine learning model. So in our training, we do have these texts. We have images and we have associated texts. So now the decoder, with the help of this attention mechanism, it learns to observe specific features of the input image and associate them with specific parts of the text on the output chemical notation and structure. And what our decoder does is that it pretty soon learns that all of our chemical, in this organic chemistry example, it starts to always predict carbons. Then it goes out into the image and searches and counts how many carbons we have. Then we have the hydrogens, the number of the hydrogens, and so on and so forth. For the decoder part of this architecture, we put together two different flavors of recurrent neural networks that predict each step one by one time, and that's the way they train as well. These are the two flavors are with GRU and LSTMs, gated recurrent units and long short-term memory units, together with back-to-now attention. But again, what I would like to emphasize is our implementation of a transformer with self-attention. And the implementation of the transformer, what it helped us do is to significantly speed up our training process by parallelizing it. And by parallelizing it using advanced linear algebra. That's an intuitive way that you can think about it. Next, and the final step is our post-processing step, in which we pass our prediction through RDKit, which is an open source Python library, and it helps us to remove invalid molecules. And what that helps us do is do a final re-ranking of our predictions. This step is helping us boost our model's performance. Next, we will discuss the success of the solution, which on top of its modularity and flexibility, as you saw, we implemented multiple different flavors of both encoders and decoders and attention mechanisms, so you can swap and use whatever is best for your particular use case. We also are proud of the cost efficiency of this particular solution. As you can see in the graph at the top right corner, the Quantiphi solution is 60 to 200 times faster and cheaper than some other state-of-the-art solutions. We achieved that by building an advanced engineering solution on top of a powerful TPU architecture with advanced optimizations, and by creating a pure TensorFlow pipeline with pretty much everything TensorFlow has to offer. Now, having a great machine learning model and an engineering solution is arguably every applied research team's goal. But as companies, what we mainly care about is how we can use this solution to increase our revenue. And the answer is no other than machine learning operations and operationalizing these models, which means operationalizing and automating the end-to-end solution for our daily use. Google Cloud provides 3 different options for orchestrating such pipelines, namely through Vertex AI pipelines, Kubeflow, and TensorFlow extended pipelines. Like in this case, most pipelines start with the data ingestion. Usually typically this is done into Google Cloud storage that can deal with both structured and unstructured data formats. Then after we've ingested our data, we have our preprocessing step, which can be handled, for example, by Dataflow, which is a wonderful serverless component of Google Cloud, as Vincent explained in detail at the beginning of the session. And Dataflow performs extract, transform, and load, or ETL type tasks, for example. Another way we can do our preprocessing is through Vertex AI notebooks, for example, for the creation of compressed TF records from our raw data sets. Then we can train our model, our custom machine learning model, with its optimizations using compute engine and cloud TPUs. These very powerful hardware that are machine learning specific. Vertex AI and cloud helps us automate our hyperparameter tuning experimentation through Vizier and training pipelines, where all we have to do is to define our metrics of choice and the ranges for the parameters we want to tune on. And then we can track our experiments through the experiment tracker or visualize them in TensorBoard. And finally, we can choose the model with the best performance as the model that we will then move forward into production. Once we've chosen this best model, the best performing model, we can then export it into production, either in Google Cloud Storage for batch predictions, or what we can do is we can deploy it at an endpoint for online predictions. Google Cloud provides a model monitoring functionality that can be used to monitor the performance of our model on new predictions. Using model monitoring, we can orchestrate a continuous training, evaluation, and redeployment pipeline that trains our model in continuously newly seen data when its performance deteriorates and drops below some acceptable threshold. We can even set alerts that let us know when our model gets below this acceptability limit. And that is an example of a powerful end-to-end pipeline on Google Cloud that can bring our models and your models from deployment straight to production while ensuring that we serve the best quality models at all times. The next model that we will discuss is our second use case for the day, and that is no other than molecular enumeration. And let's start by putting some context to that. What exactly is numerical enumeration? Simply put, it's the process of creating new molecules for drug discovery. The generation of new drug candidates is a very important step in the process of drug discovery. Currently, there are several challenges associated with it. The molecular enumeration is a time-consuming and expensive process, and as a slow process, it can lead to long research and discovery cycles, as Tim discussed in detail at the beginning earlier. Now, these enumeration workflows can also get computationally heavy, as also discussed by Vincent, and what they require is scalable solutions. So what we will aim to do, and we will discuss later today, is how we can orchestrate and put together a scalable workflow utilizing some Google Cloud components such as Dataflow and open source libraries such as RDKit that can help us speed up enumeration workflows and can help us handle billions of enumerated molecules. At the end of the day, what we care about is to produce revenue by optimizing and operationalizing your drug discovery workflows. And what that can help us is it can lead to potential savings in the drug discovery process and also time saved, which again is a very important parameter. Next slide. In general, a molecular enumeration workflow can be divided into 4 steps. Our enumeration algorithm that can generally have 2 flavors. It can either be that of a machine learning model or a library-based approach, such as RDKit, which is an open source Python library that we will talk about later on. Today, we will talk about this RDKit workflow. With the help of the enumeration algorithm, whatever that may be, we can generate orders of magnitude more molecules than the seed ones that we used at the beginning, at the starting of this process, reaching up to billions of generated molecules. Now, with this plethora of new molecules, what the next typical step is that we filter them down to only consider for the next steps of our process, those that have some particular wanted properties, required properties, such as drug likeliness, patentability, synthesizability, novelty, solubility, and diversity. Once this filtering is done, we are then left with a subset of molecules that are potential drug candidates. Next, we will discuss a particular open source library called RDKit that allows for reaction-based enumeration. Typically, a company will have a large chemical database that our scientists will query to match some molecules with specific properties, and they will also define types of reactions that we are interested in exploring the products of. RDKit takes as inputs exactly these two parameters, reactants and reaction rules between these reactants, and handles pretty much the rest. So what it does is that it simulates these reactions and produces orders of magnitude more products than our input reactants. For example, in this case, I used an open source database and searched for heterocyclic compounds, alkynes, and azides. I then defined a nucleophilic substitution reaction between 5-member heterocyclic compounds and alkynes. The product of this reaction, I reacted with through a click reaction process with the azide compounds that you can see at the top of the screen, or rather a family of them. And the final number of products downstream is what we got. And in this particular process, left me with at least more than 2 orders of magnitude more molecules than those I started with. That is 100 times increase. So with your vast databases and with your more complex chemistry knowledge than mine, with multi-layered reaction processes, it's very easy to reach very large numbers of generated molecules. Next, we will talk about an example of a modular and scalable Google Cloud architecture that can handle this workflow that is very data intensive, and we need it to be scalable. As discussed earlier on, we start by defining our reactants and our reactions that we're interested in exploring for this enumeration instance, and we ingest these into Google Cloud storage. Then our main enumeration step can be handled either by data flow as an ETL, extraction, transform, and load workflow, or by another Google Cloud component such as Cloud Run that allows us to use containerized custom code solutions regardless of the language of our preferred language programmatically. Both alternatives are scalable and they are easy to maintain once orchestrated through a pipeline, as they can scale to fit our resource needs. For example, employing a larger number of computational resources to parallelly process our data-intensive enumeration operations and shorten our drug discovery cycles, while at the same time keeping the cost into a minimum, as we only pay for what we use and we don't need to a priori allocate these resources. The last step in this process is the filtering step. BigQuery hosts some tables on molecules that have already been patented, which we want to exclude from our downstream discovery process. As discussed earlier, patentability is not the only property we are interested in filtering for, and we can orchestrate further filtering steps either through Cloud Run and RDKit or through, for example, Vertex AI Notebooks. The final product that we get at the end of the day is that of non-patented molecules with specific wanted attributes and properties. What I want to emphasize here is, while in this particular case we do use RDKit for this pipeline is a modular one and can handle any enumeration algorithm you are using for your day-to-day workflows. And with that, thanks for your attention, and I'll pass it on to Vishal that will talk about drug effectiveness next.
Yeah. Thank you, Sophie. Okay, so going with the story so far, so we talked about how within your research papers, which you have in your database, how you can extract the molecule information using machine learning models, and then we move to how you can create your compound library from by using the molecular enumeration pipelines, right? So from there now we move to the next step, which is to try and understand how you can use your compound library that you have generated against any protein of interest. And here we are specifically going to talk about COVID. Like Vincent mentioned at the start, so this effort which we did was to basically understand how as the COVID virus is mutating and then evolving, what kind of trends we can expect from that, and then how do we have a framework to actually understand the kind of effect which it might have in a real-time basis, and then be more proactive and then try to act accordingly. So in this, so we have 3 basic tools involved, and then three basic like functions, right? So what we wanted to do was now you have your COVID spike protein, you know that the virus is mutating, so you want to understand or you want to have a pipeline which gives you all the possible future mutations, right? Or at least a framework which tells you like how the virus can mutate and then what its at least a primary sequence is going to look like. So that's a mutation generation pipeline. And once you have your primary sequence, this is where we plug in AlphaFold. So we know that AlphaFold has really good accuracy when it has to generate 3D structures of proteins, and that's something which is enabling a lot of research within the life sciences community. So and using it is as simple as just like passing in a FASTA format. So we pass in the FASTA file to AlphaFold, and then basically we get the 3D structure. So we are using AlphaFold to generate the 3D structure for any mutant protein. And then we move to screening, and we use virtual screening to understand what the differences are, right? So what happened before, and then what kind of changes does it present so far. So with that, we'll move to the architecture and then talk about how we have done this or we have implemented this. So we basically started working with the Gizzard dataset. So we obviously, like, it's a large dataset. So we do not use like most of it. We just sampled like a few sequences of different variants. So we used the original spike protein, which serves as our baseline, and then we used the Delta variants, the Omicron variants, and like I said, the first setup or the first step for us is to generate these mutations, and we limited these mutations to a lot of substitution type mutations and not any insertion deletions, which could have like slightly different dynamics when you talk about like the evolution over time for a virus sequence. So we generated these mutations, and that's when we used AlphaFold. And usually AlphaFold gives you like 5 structures. So it gives you the best 5 structures, and the top one is supposed to be the best one. So you can use that. But when you're actually implementing your own pipelines, because AlphaFold gives you the way to understand its confidence at different levels or at different parts of the 3D structure. It doesn't hurt to actually use all the 5 sequences in different ways and then try and run those pipelines. So we take the 3D structures, and then the first step was just to visualize how it looks when compared to the original spike protein. And then we moved to the protein docking paradigm. So what we did is we basically screened it against the EC2 protein and also different antibodies which we could like shortlist at that point. And the idea is to understand the binding energy difference between a mutant and the baseline or the original COVID protein, right? So as the mutations, as the COVID virus is evolving, you want to understand whether it's getting better or worse at binding to ACE2, which becomes the first line of entry for it, and then also antibodies, which are your first line of defense, right? So we use ClusPro to basically get these binding free energy differences, which is with antibodies and ACE2, right? We did the same thing with Autodoc, where we used like small molecule drugs, and we did similar things. So 3D structure from AlphaFold, take the PDB files from the protein data bank, and then we did a virtual screening again on Autodoc, and we eventually like got them together. And what we did is we compared them. We compared the samples we have with different tools. So as you can see, there's another tool here called PPAPRED, which is which it does not use 3D structures. It just needs the primary sequence, and it still gives you like an affinity prediction. We just compared both of them, and the patterns still hold. So whichever sequence or whichever variant showed that it had like a better affinity towards ACE2 protein. We saw that the results were similar when we also check a different tool. And once we generated like sufficient amount of like results where we know the variant and the effect of the variant, which is like better or worse, which is binary, and the types of mutations it had and then where it had, we built a simple machine learning model like down the line where we use that to predict its effect. And it turns out like even though the data was not like that scale, we were actually able to like replicate the 3D docking studies and then have a model which can actually predict it with the information which has been annotated. So this is the overall structure. Now we'll move to the next slide where we see how we implemented this in context of the Vertex AI ecosystem. You can see Vertex AI is the foundation for all the work which we did here. Most of the experimentation and implementations also happen on the Vertex AI notebooks, which turns out to be a very handy tool, and also it can give you ability to use GPUs, so it does not come with a small scale. You can actually run decent sized experiments on notebooks. So we actually used Vertex AI notebooks to generate a 3D structure. So we basically spun up a large instance for on Vertex AI notebooks, and that was able to download all the database that AlphaFold usually requires. And we were able to generate the 3D structure within the Vertex AI notebooks. And we also used that framework for other things here, as you can see, for mutation generations, and also the machine learning model which we built, and also the screening studies with Autodoc. Because here we were conducting a feasibility study, we wanted it to be very interactive, so we used the notebooks. But once you start thinking of automation, that can easily be replaced by any other tools like either Google, the Kubernetes engine, or Cloud Run, to make sure that you are actually having triggers to run these in an automated fashion. You can also see another tool, BigQuery, here, which can help you if you once you have structured data and then you bring them together from screening. And like I said in the last slide, we built a machine learning model which was able to tell us, or was able to replicate the docking results from a machine learning standpoint. So with BigQuery, you can use BigQuery ML, which is a handy tool which comes within the BigQuery ecosystem. So you can train these models by staying within the tool, and you don't essentially have to do a lot of coding and then build these models. Yeah. So with that, we'll move to the last slide just to summarize the results. So you can see we had quite a few pipelines, right? And all of these were actually run on GCP within the Vertex AI ecosystem, except for the structure-based protein screening, which was the ClusPro tool, which was a web-based tool. So we didn't essentially control the servers, but we using a web tool there, which can easily be replaced by any open source tools, and all of that can be implemented on GCP in a scalable fashion. And like I said, this was a feasibility study, so we built a framework for the client to make sure that whenever a new variant comes in, all they have to do is pass it through AlphaFold, get the 3D structure, and they can pass it through different scoring algorithms to understand whether it is whether you can predict it to have a better effect or a worse effect when compared to COVID in the grand scheme. With that, I'll leave it back to Tim.
Thanks very much, Vishal. So the good news is that the market is evolving, and we are seeing some positive trends and movements that are indicative that AI is becoming an adopted type of approach to be able to solve some of these intractable problems that we talked about at the onset of this call. So far, over 50 FDA approvals have come forward, or been approved by the FDA, I should say, for machine learning and AI algorithms, life sciences. That's positive. Gartner is saying that by 2025, about 30% of all new drugs will be developed using AI ML capabilities. And the other piece, from a leadership perspective, indicates that generally leaders are proponents of these types of approaches. Over 90% or so have indicated that this is going to help in the development of drug discovery. On the other side, there's also the impacts and how we might be able to best use AI ML capabilities within the overall value chain for pharmaceutical manufacturers and life science researchers. Specifically, obviously, we've talked a little bit about the drug discovery pieces. In silico drug discovery and testing is a huge area of opportunity for AI ML. Also, the ability to integrate knowledge graphs, particularly as you're sifting through all the information that's currently available to identify different molecules, as an example. And drug trials, although that the completion rates are pretty low today, being able to use ML capabilities to identify better patients that should be qualified in a particular study, in particularly those patients that are going to complete the studies are going to be an important part of that overall approach. Predictive modeling to be able to identify, what those drug study completion rates will be. We're also seeing things like digital twins as an example in this particular area as being another way of being able to reduce perhaps the number of patients that are needed, but being able to increase compliance and completion rates. The other piece, of course, is around the manufacturing and distribution. Particularly when we're taking a look at, for instance, like sentinel events for adverse events, beginning to identify which patients are going to be at risk earlier in the process are part of the AI-assisted quality control mechanisms that we're looking at, as well as, of course, looking at demand forecasting for the particular types of agents, reagents, as well as the molecules that are going to be needed to be able to support these types of drug discovery implementations. So a lot of exciting stuff happening here, and we're optimistic, obviously bullish on the opportunities as an AI company, and we think that this is an area of ripe opportunity. Anyway, from there, I'm going to pass it over to Vincent to wrap us up and take any questions.
Hey, Tim, Sophia, Vishal, fantastic. So at this point, we're going to take questions. You can either unmute, raise your hand, put a question in the chat. I'm going to leave this slide up here now. If you have any questions for me that come up after this call, or if you have any recommendations for one of our future seminars, you can reach out to me at vjbeltrani@google.com. And if you have any questions or you want to explore any of the solutions that Quantiphi presented today, you can also reach out to Harshini Infanta, the director over at Quantiphi. So with that being said, we have an upcoming webinar. We're bringing in another partner, Ontochem, who has produced a search engine for chemistry and chemical information that's even more powerful than tools you use today. Kind of like you probably use SciFinder, and I think you'd be very impressed to see some of the tools out of the Ontochem team. All right, with that being said, does anybody have any questions on any of the material that we covered or anything that they want to discuss with me or with the Quantiphi team?
So Vin, I think there's a question in the chat from Jake Siegel.
Let's see. Good question, Jake. So is synthesizable prediction provided by Google? So we have collections of Savvy2. So there are databases, publicly accessible, and databases that we host that provide you with synthesizable or 1 to 2 step reaction synthesizable compounds. We also could work with you to cross-reference maybe your compound libraries or the compounds that you have access to help filter down those steps when we go and generate those large virtual libraries, for example. We could definitely follow up with that, Jake, as well on one of our calls. Let's see.
Yeah. And Vincent, there was one more question. How can we connect Google Healthcare API with Vertex AI?
Oh, that's a great question. So the beauty of our healthcare APIs is that they're all API driven, and directly from one of your Vertex AI pipelines, you can either leverage them through their API calls, or you can use the healthcare API or healthcare data engine and those kinds of tools ahead of time to parse your data, put it in BigQuery, and then make it available for anyone running any machine learning pipelines on Vertex AI. So you can most certainly include all of that ecosystem of tools, our NLP API, our healthcare API, all of that directly inside of some of your machine learning pipelines. All right. Are there any other questions for the team? Feel free, like I said, to follow up with me. Also, please feel free to reach out to Harshini. Thanks, management must be able to connect those agreements hard to achieve. Oh, that's definitely something that we can follow up on. So why don't you reach out to me? You send me an email, and that's a conversation that we can certainly have. But I don't know if it's hard to manage. Some of the nuances really are just kind of learning a little bit more about how to set up things like IAM, because Google Cloud has comprehensive security features. And it really could actually be a very easy problem to solve as long as we just approach it the right way from the beginning. So let's have a follow-up call on that one for sure.
And Vincent, there was also a question by Hitesh about are there any R&D use cases for generic pharma manufacturers?
Hitesh, are you online still?
I
was
think he
just kinda
just
curious.
left.
Oh, it just left? Okay. So then we'll follow up. We have the I'll follow up. Are there R&D use cases for generic pharma manufacturers? Sure. I think in the sense you say generic pharma manufacturers, presumably that's maybe follow-on drugs or drugs like that that have already been published or that are already available. You can have access to clinical trial data. So we have tools to give you access to clinical trial data. If you think you can make a better molecule or a generic version of a molecule, could also help you out doing things like that. So I know Hitesh is on the line, but Hitesh, we will find you and reach out to you, and we will have a follow-up conversation about exactly that. All right. Well, if there are no more questions, with that being said, really thank you for your time and for your attention. We're looking forward to this webinar series. It's going to be a very science-focused webinar series where we'll be bringing in our partners to describe the solutions like you saw today. We'll talk about the science, and then we'll also talk about the architecture on Google Cloud, motivating our customers to leverage things like the managed services that I talked about, Vertex AI, talk more about BigQuery and how to build chemistry or cheminformatics using BigQuery tools.